At this time, I'd like to turn today's program over to Ingrid Miller. Ingrid, the floor is yours.
Thank you, Emily. I'm a product manager with the Biotechnology Explorer program, and I wanted to thank all of you for joining us for the webinar today. We also wanted to thank you for providing the pre-registration feedback that you provided to us. What we know is that we have a nice cross-section of audience with attendees from the high school, community college, and university segments. So thank you for taking the time to provide that feedback ahead of time. And right now, I'd like to introduce our speaker today. Jim DeKloe is our speaker, and he has been a professor at Solano Community College in the North San Francisco Bay Area since 1990, and he currently serves as the director of the Industrial Biotechnology Program there. This program emphasizes biomanufacturing by training students in the skills required to work in companies that are located in the biotech manufacturing cluster in Solano County, which is situated between San Francisco and Sacramento. Jim also serves as the regional director of the Southwest region of BioLink, which is a nationwide consortium of community and technical colleges that teach biotech. During a year-long sabbatical, Jim worked in the manufacturing department of the biotech pioneer Genentech Incorporated, gaining in-depth knowledge of real-world biomanufacturing processes and needs. He has also served as a consultant to help the biotech giant Amgen redesign the training programs for their biomanufacturing technicians and for their quality assurance associates. In 2000, the Association of Community College Trustees chose Jim as the distinguished faculty member of the Pacific region, which includes the Western Continental United States, Hawaii, Alaska, Canada, and Guam. Thank you, Jim.
Thank you for inviting me today. Our lab has been beta testing a new experiment that Bio-Rad developed, and we'd like to share our experience with you today. Today, during this webcast, we'd like to look at expression in different cell systems, look at this experiment and how it helps students teach protein recovery and purification, talk about different types of chromatography, and then in the end, what I always do with students is I try to relate to them how the small-scale laboratory experiments relate to the way that biotechnology companies manufacture protein pharmaceuticals, the way that they scale up to a giant scale and produce kilogram levels of protein. First, I'd like to talk about the biotech industry, and the way that I view it is this is a lot like alchemy. The ancients tried to produce gold by converting base metals like lead into gold. And what biotechnology does is it takes materials that are not worth very much money, mainly sugars and salts and water in the media, and by using genetically engineered cells, it converts those to products that are much more valuable than gold, that are worth many, many times their weight in gold. And so on this slide, if you look at the current price of gold, which is high right now, many of the products, like insulin, is worth more than its weight in gold. Growth hormone is worth many, many times its weight in gold. And then one of Amgen's products, granulocyte colony stimulating factor, is worth about $1 million a gram. And what I do to students is I remind them that a penny weighs about 3 grams. And so a gram is about 1/3 of the weight of a penny. So if they had a penny's weight of granulocyte colony stimulating factor, they'd have a product that's worth more than $3 million. What biotech companies do is they take genetically engineered cells and they produce proteins, and I always remind students that proteins are the product. It's proteins that are injected into patients. In some future time, there might be interfering RNAs, there might be DNA therapies, but currently biotech is protein pharmaceuticals. Protein is the product. And what the biotech companies do is they take DNA and genetically engineer the cells, and then this is a good time to review the central dogma. Central dogma is a term that was coined by Francis Crick, and he later on admitted that he didn't really know what the term dogma meant, but we're sort of stuck with the term. So information flow goes from DNA to RNA to protein, and then proteins are the tools of the cell. proteins fold in a particular three-dimensional structure, and that three-dimensional structure then carries out the function of the protein, which, whether it's an enzyme or a structural protein, will produce a phenotype, produce a structure, produce a characteristic of the organism. For a long time, our lab has used the green fluorescent protein system of Bio-Rad, and this I've found to be an excellent teaching system where you use the pGlow plasmid carrying the green fluorescent protein gene, transform that into E. coli, and it's been engineered so that the enzymes within E. coli will recognize the gene, transcribe it to form an RNA. The RNA then will go to ribosomes and direct the synthesis of the green fluorescent protein, which then makes the cells fluorescent. I've always worried, though, that this gives students the wrong impression. This gives the students the impression that protein always is visible, that protein always glows. And so this new system takes a gene for an enzyme, dihydrofolate reductase, and uses bacteria to produce that human protein. And again, the dihydrofolate reductase gene is in a plasmid, it's in a bacterial cell. It's been engineered so that the bacterial cell will produce messenger RNA. The messenger RNA will go to ribosomes and direct the synthesis of the dihydrofolate reductase enzyme. The dihydrofolate reductase enzyme we'll talk about later is really important in nucleotide synthesis. Again, the products of biotechnology are proteins, and this slide shows some of the examples of proteins that have been engineered to use in humans. The first protein that was produced using genetically engineered cells was insulin. Insulin was an important protein because before the development of this technology, insulin was collected from slaughterhouses. Eli Lilly, especially in Indianapolis, collected pig pancreases from all over the United States. They'd grind them up and they would isolate the pig insulin to inject into diabetics. It worked, but there was one amino acid different out of 51 from human. And the production of pigs was going down in the United States as the number of diabetics were going up, and they projected a shortfall. They bought the genetically engineered cells from Genentech, and then all insulin all around the world now is used using genetically engineered cells. So the genetically engineered cells have the human insulin gene, and they're tricked to produce human insulin. The next protein that was made, again by Genentech, was human growth hormone. Human growth hormone is produced in the pancreas. Again, there was a severe shortage of human growth hormone by putting the gene into E. coli. E. coli then produced human growth hormone, which can be used to treat people who, without the treatment, would be very short in stature. Another important product is Factor VIII. Factor VIII is the missing clotting factor in the most common type of hemophilia. Hemophilia is a really serious disease. If you think about the inability to clot, every cut, every bruise presents a life-threatening situation. Hemophiliacs then, taking Factor VIII, have normal clotting for their blood. A huge product, $8 billion a year, is human erythropoietin. Human erythropoietin is used to treat people who are undergoing cancer chemotherapy, whose ability to produce red blood cells has been compromised, and also people who are undergoing dialysis, who usually cannot produce enough red blood cells. This makes a dramatic difference in their life, not only giving them more energy by elevating the number of red blood cells, but it also has an effect where it often alleviates chronic depression because your mind and body is so closely linked. One of my favorite proteins is tissue plasminogen activator, pioneered by Genentech. This is an enzyme that digests blood clots. And by digesting blood clots, it can be used to treat heart attacks and strokes. The most common type of heart attack, a myocardial infarction, is a blood clot in a coronary artery. As that blood clots, it cuts off circulation for the entire heart tissue downstream of the clot. by injecting tissue plasminogen activator, it goes right to the clot and specifically digests the clot, restoring blood flow, preventing the damage to the heart tissue downstream of that. Also, the most common type of stroke is a brain attack. It's a blood clot in the arteries of the brain. When a clot blocks the delivery of oxygen and nutrients to the brain tissue begins to die relatively quickly. If you can get the patient tissue plasminogen activator within the first few hours, it'll digest the blood clot, restore blood flow, and that part of the brain then will not be compromised. That part of the brain will not be destroyed. Granulocyte colony stimulating factor is a protein that stimulates the production of white blood cells. And this is used to treat patients who are undergoing cancer chemotherapy. Cancer chemotherapy also, we talked about it before, diminishing the production of red blood cells. It also will compromise the production of white blood cells in the bone marrow. White blood cells are an important component of the immune system, and without white blood cells, a devastating septicemia, bacterial infection, will almost always follow when particular white blood cells fall below a certain level. By coupling granulocyte colony stimulating factor injections with cancer chemotherapy, then the patient will not experience these deadly septicemias. You can see there are other proteins, and we can talk about those at some other time. I wanted to talk about how you produce these proteins, and there are several choices of which type of cell to genetically engineer. Most people are familiar with genetically engineering the bacterium E. coli. Escherichia coli has been an important model system since the 1930s, and when it came down to choosing an organism to develop recombinant DNA technology in the early days of biotechnology, E. coli was the logical choice. And then E. coli was used by biotech companies in scale-up efforts, and still many, many products are produced by growing E. coli at the large scale. But there are other systems. Some biotech companies use yeast, and this is the typical yeast, brewer's yeast or baker's yeast, Saccharomyces cerevisiae. It can be genetically engineered. It has a few advantages since it's eukaryotic. It has a nucleus. But many, many biotech companies use mammalian cells, and the mammalian cell of choice is a Chinese hamster ovary cell. And that choice was made mainly for a regulatory consideration rather than a scientific consideration. And we'll talk a little bit later about why CHO cells were chosen, and it has to do with this enzyme DHFR. If you look at the advantages and the disadvantages of these different systems, you will find that the organism of choice is typically E. coli. If you can get away with genetically engineered E. coli to produce your product, that's what you will choose, because it grows fast, it produces a lot of protein, the cost of the medium is relatively inexpensive, and because it's growing fast, the risk of contamination is relatively low. So this is a good system that you can use if your protein does not have to be glycosylated, that is, if it isn't important to covalently add polysaccharides to the protein, and if the E. coli can fold the protein properly. Remember that proteins, in order to function, have to have an appropriate three-dimensional shape. They have to be folded properly. E. coli has trouble folding very large or very complex eukaryotic proteins. If it can't fold the protein, then you either have to unfold and refold the protein or choose a different system. Yeast has some advantages, although the early promise of yeast has not been fulfilled. This is a system that has far fewer applications than anyone predicted, although the applications that it does have are huge, and we'll see that on the next slide. Mammalian cells, that is CHO cells, are used very common if your protein doesn't fold in E. coli and if it has to be glycosylated. But the cost of the medium is very high. It grows relatively slowly, so you have a long window for contamination by exterior bacteria, and one bacterial cell, you have to throw away the batch because bacteria grow so much faster than the CHO cells themselves. Their division time is much shorter than the CHO cells. Chinese hamster ovary cells will properly fold the proteins, and if glycosylation is needed, this is the protein of choice. But it pays off because often during the protein purification process, it makes it a little easier, and often CHO cells have been genetically engineered to take the protein, make it on rough endoplasmic reticulum, and then secrete it into the media surrounding the cell. Therefore, you can centrifuge or filter out the cells and keep the harvested cell culture fluid. On the next slide, it shows some examples of some of the products and what the organism of choice is to produce the product. Insulin, being a really short protein, only 51 amino acids in the final product, is produced in E. coli. Human growth hormone folds properly in E. coli, so that's the organism of choice. Granulocyte colony stimulating factor, again, that's the protein that increases the production of white blood cells. That's produced in E. coli. Tissue plasminogen activator protein, on the other hand, is a very large protein, 527 amino acids, really complex, many domains, difficult for E. coli to fold, and therefore the cell of choice is CHO cells. Also, tissue plasminogen activator has to be glycosylated. It has to have these carbohydrates covalently bonded to it, or else it is cleared by the liver quickly from the bloodstream. And you can alter the half-life of a protein in the bloodstream, and Genentech has done that, by altering the glycosylation pattern. Pulmozyme is an interesting product that we haven't talked about before. This is a protein that digests DNA, and it's used in the treatment of cystic fibrosis. Individuals with cystic fibrosis, this inherited disorder, which is relatively common in the United States, build up a thick mucus that's very viscous. And part of that viscosity is DNA. They inhale pulmozyme. Pulmozyme degrades that DNA into nucleotides. That diminishes the viscosity of the mucus from syrup into a more watery state, and therefore it can be coughed out much easier. There's a funny story about the clinical trials on pulmozyme. They had a double-blind test where neither the physician nor the patients knew who was getting pulmozyme and who was getting the placebo. And so while the parents and the physicians were talking, the kids went out back and they were spitting on the side of the wall, and the loogies that flowed really quickly, aha! Hey, you have pulmozyme, and those that stuck at the top were placebo, so the kids found a way to overcome the design of the clinical trials. Human erythropoietin is a really important product. Again, produce over $8 billion of sales per year. This product is produced in Chinese hamster ovary cells because it's large and complex and it has to be glycosylated. If you received a vaccine against hepatitis B virus, that vaccine has been produced in yeast, Saccharomyces cerevisiae, brewer's yeast. Likewise, the vaccine against human papillomavirus. I wanted to mention antibodies. Herceptin is an example of an antibody, and antibodies are large, complex molecules. Because they're large and complex, they're made in mammalian cells. Herceptin is an interesting example. It was the first antibody that was humanized, that had human protein sequence in it. This is an antibody against a protein that's on the surface of an especially aggressive form of breast cancer. This antibody will bind the breast cancer cells specifically and lead to their destruction. Before Herceptin was marketed, women with this aggressive type of breast cancer were given 9 months to a year to live. Now, women under Herceptin treatment, along with traditional chemotherapy, will live past the 5-year benchmark. I always tell students that the genes that are on the plasmids that they're transforming the cells are always placed behind an inducible promoter that allows you to turn the genes on and turn the genes off. Biotech companies always genetically engineer their plasmids so that they have control of whether or not transcription takes place. You're probably familiar with the pGlow plasmid that has green fluorescent protein. In the pGlow plasmid, the green fluorescent protein has been cloned behind a promoter that's been taken from the Arabinose Operon. This allows you to turn green fluorescent protein production on by treatment with the 5-carbon sugar Arabinose and keep it turned off before the Arabinose has been added. This sort of ability to regulate the production of messenger RNA and therefore the production of protein was first worked out in the Lactose Operon by the French scientists Jacob and Monod. As a review, the Lactose Operon is 3 genes that are involved in the breakdown and metabolism of lactose, and those 3 genes are controlled by the binding of a regulatory protein, the Lac repressor. In the absence of the disaccharide lactose, Lac repressor is bound, and therefore the RNA polymerase has no ability to access the promoter, and therefore the gene is turned off. In the presence of lactose or its analogs, the lactose binds to the Lac repressor, causing it to be removed from the DNA. Therefore, RNA polymerase has access and the genes are transcribed. What biotech companies can do is take the genes for the Lac operon, remove them, and put their gene of interest there. If the gene of interest is regulated in this way, if it's cloned behind a Lac promoter, then analogs of lactose can turn the gene on or turn the gene off. In the system that you may be using for green fluorescent protein, it uses the Arabinose Operon. The regulation of Arabinose is slightly different, although it involves a regulatory protein. This regulatory protein, in the presence of Arabinose, turns the gene on. And so only when Arabinose is present can the ARA-C regulatory protein bind to Arabinose and turn the gene on by allowing RNA polymerase to bind. And what has occurred in pGlow is that these Arabinose Operon genes have been replaced with green fluorescent protein. And this biotech companies always do. They always have the ability to turn genes on and turn genes off. And the reason for this is that when they grow large volumes of protein, they always use 2 phases of growth. In the first phase, they grow cells with the genes uninduced. They grow cells in such a way that the genes aren't producing any recombinant protein. They've devoted their entire energies into producing more cells. As the cells start to enter stationary phase and stop dividing, then the inducer molecule is added. Production of the genetically engineered gene protein is induced, and then the cell makes a transition during the stationary phase to devote its entire effort into producing the protein of interest and filling up with that protein in anticipation of you collecting the cell The cells are collected either by filtration or by large-scale centrifugation, and then they're broken open. There are two steps. Recovery refers to the separation of the protein from the other molecules of the E. coli or other cell. So in recovery, the protein is separated from the cell walls, from cell membranes, from the DNA, from everything except protein. Proteins are liberated, and they go on to a purification step. Purification is separation of your protein of interest, your drug, from the other proteins that are in the E. coli cell, and there'll be a lot. The best systems will probably produce about 20% of the protein in E. coli as your protein of interest. And you have to get rid of that 80%, and the FDA demands that you isolate your protein to 99.99% purity in order to inject it into patients. Remember that these proteins may be injected into children, and therefore having pure proteins is very important. The proteins are separated using mainly chromatography methods. In chromatography, you always have a mobile phase, a solvent that's traveling over the stationary phase, which is usually a bead of some type that has been covalently modified with a group of that's been covalently bonded to it. Molecules travel in the mobile phase through the stationary phase, and then they'll interact to a greater or lesser extent with the solid phase, and that serves as the way to separate the proteins. So, for example, in size exclusion chromatography, a chromatography system that separates proteins according to their size, a mixture of proteins is added to the top of a column. That mixture of proteins flows through the column, being carried by the mobile phase. Small proteins will interact more with the solid phase than large proteins, and therefore large proteins will move faster, and the large proteins will be able to be separated from the smaller proteins. Other types of chromatography systems will use other characteristics of the protein in order to separate those proteins. Again, size exclusion, the other name for it is size exclusion separates gel filtration chromatography, separates proteins according to their size. Ion exchange chromatography, which is used at the large scale a lot, separates proteins according to their charge. As you know, proteins might be positively charged, negatively charged, or they might have a neutral charge where the positives and negatives are balanced, and these will select proteins according to that charge. Hydrophobic interaction chromatography separates proteins according to their hydrophobicity, according to the ability of their amino acid R groups to interact with water. And then affinity chromatography chooses a special property of that protein to separate it from other proteins. For example, protein A is used a lot in biotechnology because the separation of antibodies from other proteins is so common. Protein A is a bacterial protein that's been genetically engineered and produced in large quantities that will be used as the first step in any antibody production method to grab onto the antibody molecule and have the other proteins flow through. This is effective, and it's going to isolate the protein to 90–95% purity right away in the first step. There's also a system where you can add several histidines, usually 6, covalently bond them to the end of your protein. Proteins that are his-tagged, that have these extra histidines, can go through a column that has nickel, and those tagged proteins will be grabbed, and other proteins then will flow through. There's a similar system with glutathione S transferase. This is an enzyme that is fused to your protein of interest, and again, there's a special affinity column that will grab that. You may be familiar with the hydrophobic interaction chromatography columns that are used to separate green fluorescent protein. I always use these as the first step, as the chromatography system that I introduce the concepts of chromatography to the students. They can see the fluorescent, green fluorescent protein move into the column, they can see it bind, and they can see it displace when you change the mobile phase to the elution buffer. And so green fluorescent protein is an excellent teaching method to teach these principles of chromatography. Usually a biotech company will use multiple chromatography steps in series. So if there's an antibody, the first chromatography step will always be protein A, and this is really expensive. The protein A columns are large, thousands of liters are put through them. They grab protein A, and they are, the column probably contains $1 million of protein A resin. Again, that will isolate proteins to a 90-something% purity, but that's not good enough. And so the protein will be subject to exchange columns that will separate the proteins according to their charge, maybe a hydrophobic interaction column that will separate proteins according to their hydrophobicity. And by having separate columns that exploit different properties of the protein, that's where you get your purity. You separate the protein according to a special property, and then according to its charge, and it's relatively unlikely that a contaminating protein will have that special property and the same charge, but then if you use another column that separates according to a different property of the protein, a hydrophobic interaction, it's going to be really unlikely that a protein will stick to the protein A column and then have the same charge and have the same hydrophobicity. If you add a chromatography column that separates according to size, then it makes it really unlikely. Affinity chromatography can also be used to grab your protein and then change the media surround it, and that's often used at the small scale. It's used to desalt a protein solution to change it from a salty solution into a low salt buffer that surrounds it. When you perform chromatography, I always have the students, before they move on to any automated system, I always have them do it old school. We all had to do it, and so I think as an initiation, they all have to do it. They have to hand pack their columns rather than buying a commercially prepared column. They have to add their sample by hand, and they always have to, in my lab, count drops using gravity chromatography rather than having a pump it through. I like it because it gives them a better idea before they move on to some other chromatography system. So cells are often broken open, and then you can use a step for the chromatography. On the laboratory scale, you can have gravity feed the protein through the chromatography system, or assist gravity by putting a G-force behind it, and a lot of small-scale chromatography systems have you put it in the centrifuge and have a spin column. I always like to then, if you have it available, have the students move from hand packing their chromatography column and adding the buffers by hand into some sort of instrumentation, into an automated chromatography system. And I'll have them do it at our small laboratory scale, and then describe how biotech companies do this at the large scale. They manufacture proteins at the kilogram level in biomanufacturing, growing them in large bioreactor tanks, and then putting them in really large-scale chromatography columns.
So we'd like to take a moment again to remind you that if you have questions during the presentation, please send it to the presenters via the Q&A function on the right hand of your screen. If you're having any technical issues, please use the chat function to the host. And again, we wanted to remind you that this presentation and the PowerPoint and a recording of the presentation will be sent out to you via email following the presentation. Thank you.
I always start with green fluorescent protein, which I always think is a really good system for the students to use. The system works where the green fluorescent protein lights up, and it's a great color. When I first saw this, I ordered a kit from Bio-Rad, and then I transformed bacterial cell for myself before I introduced this to students, and I was wondering how dramatic would be the effect, how much would the cells glow, and I was just pleased and shocked at just how green those things are, and just how well the fluorescent element of it works. But I always worry that the students are getting the wrong impression of a protein. Most proteins are invisible. They have a color, but the color is in the ultraviolet and can't be seen. So when you work with protein at the laboratory, or when you work with the large-scale production of proteins as a biotech product, they're invisible. The buffer solutions that carry them are indistinguishable from water. And so what I've always looked for was another system to follow up green fluorescent protein that gives a better, more accurate view of how the students will encounter most proteins. And this system works well. The dihydrofolate reductase gene is in E. coli. It's inducible, and with the lactose analog, IPTG, the bacteria produce the dihydrofolate reductase enzyme. It has an advantage of being an enzyme because you can detect it using an enzyme assay. So this is more accurately the way that the students will encounter a protein. The solutions that carry DHFR protein are invisible, are indistinguishable from water. They represent the real world better. And so I always start with green fluorescent protein for fun and to give them the idea of the concepts, but then move into a protein that more accurately represents the real world situation. I want to talk a little bit about this protein, dihydrofolate reductase, and it's an interesting enzyme that has a long history and an important history that can be integrated into a lesson plan. The protein here is human dihydrofolate reductase, and what it does in human and other mammalian cells is it converts dihydrofolate into tetrahydrofolate, and it uses the electron donator, NADPH, in order to do that. The product of this reaction that's pictured there is an important methylator, and it's used in biochemistry to add a methyl group to any molecule that needs to be methylated. And the production of nucleotides requires methylation. The production of some of the amino acids require methylation. And so this is a key enzyme that is important in all mammalian cells. This enzyme proved to be important in the clinical setting as well. There are some clinical deficiencies that are caused by a deficiency in dihydrofolate reductase. And also, this enzyme was important in the early history of cancer chemotherapy. The first chemotherapy agents were introduced to disrupt this enzyme. And again, if you think about it, this was a logical step that cancer cells are growing uncontrollably. And in order to control cancer cells, you want to starve them for nucleotides so they can't divide. Nucleotides must have this enzyme to be produced, and so when the enzyme is disrupted, nucleotide production stops, DNA synthesis stops, and cancer cells stop. So one of the first cancer chemotherapy agents was methotrexate, which is an inhibitor of this enzyme. This was pioneered by Sidney Farber, who was working with leukemia, the cancer of white blood cells. You can imagine the difficulty of early physicians that were trying to develop a treatment for leukemia. Cancer therapy before that was cutting out the solid tumor. If there was a lung tumor, the surgeon would go in and cut it out. If there was a liver tumor, the surgeon would go in and cut it out. Leukemia, which is distributed all through the bloodstream, doesn't give a target for treatment, and therefore Sidney Farber thought that you could treat it chemically, and he introduced methotrexate. This remains an important chemotherapy agent today. It inhibits dihydrofolate reductase, and again, as we said, by inhibiting that enzyme, methylation doesn't occur. Since methylation doesn't occur, nucleotides aren't produced. Since nucleotides aren't produced, DNA isn't replicated, and therefore the production of tumor cells stop. Now, in a really interesting observation, after working with this molecule for years, physicians would encounter tumors that would arise that were methotrexate resistant later in the treatment. And when they examined the reason that the tumors were evolving this methotrexate resistance, they found out that the tumors had amplified their DHFR genes. They had made many, many copies of the DHFR genes, and with those additional copies, they produced a lot of DHFR that was overwhelming the ability of the chemotherapy agents to inhibit the enzyme. And that observation has been exploited in biotechnology. DHFR is important in the history of the development of the Chinese hamster ovary system. And as a matter of fact, this is the reason why Chinese hamster ovary cells were chosen as the mammalian cell of choice to produce biotech products. And by doing that, let's go back to E. coli. If you transform E. coli cells, and you do this with maybe pea-glow, you're used to telling your students that this transformation process is really inefficient. Some of the cells will pick up the pea-glow plasmid. Most of the cells will not. And you need a selection system where the cells that don't have pea-glow are killed, and the cells that have picked up the plasmid containing your gene of interest, green fluorescent protein gene in this case, will survive. And you perform that reaction by having also on the plasmid a gene that will digest ampicillin, beta-lactamase, that will destroy penicillin and its derivatives. So if you grow these bacteria in the presence of ampicillin, those bacteria that have picked up the plasmid will survive. Those bacteria that you're not interested in, that did not pick up the plasmid, will not. In the mammalian cell system, CHO cells that have been, that are DHFR minus were engineered, and you transform them with your gene of interest right next to the DHFR gene. So the transformed cells receive a cassette of your gene of interest and DHFR. Then you incubate them in a media that doesn't supply external nucleosides, and therefore the only cells that survive will be cells that have picked up your gene of interest DHFR cassette. Those cells that were not transformed do not have DHFR and therefore will die through being starved for nucleotides. So it's comparable to the E. coli system. In E. coli, only those cells that pick up the ampicillin gene on the plasmid that contains your gene of interest will survive. In CHO cells, only those cells that pick up the gene of interest DHFR cassette will survive and divide. Incidentally, what I said before can be exploited. So if you treat these cells with methotrexate, then the cells will be induced to amplify the DHFR, and by doing so, they amplify the DHFR and the gene of interest. And so these genes, which are integrated directly into the chromosomes of the CHO cell genome, these genes will have many, many, many copies of tissue plasminogen activator or pulmozyme gene or erythropoietin gene, and therefore will produce more protein, a higher titer of the protein, and secrete that into the environment surrounding them. The DHFR protein that I've been working with, beta testing with Bio-Rad, has been engineered to have a his tag, 6 extra histidines, on the carboxyl end of the protein. And it's also been engineered to have this enzyme, GST, on the amino end of the protein. So it has both of these selective systems for you to choose from in order to grab it in an affinity chromatography situation. Now that I'm showing this nice, beautiful three-dimensional structure of a protein, I wanted to remind you that I find it useful to grab protein structures from the protein database and to show those to students and have them manipulate those images. They can rotate them, they can highlight hydrophobic amino acid residues and show that those hydrophobic amino acid residues are mainly on the inside of a globular protein. Hydrophilic amino acid residues are mainly on the outside. You can highlight the disulfide bridges, and so this is a really useful tip to show protein structure to students. Back to DHFR. Again, it has a his tag here that can be exploited to grab that protein out of a mixture of proteins in an affinity chromatography system. And the affinity chromatography system that is used to grab proteins that have been his tagged is a resin that has a covalent attachment that interacts with nickel. The nickel will interact with multiple histidines to grab onto the protein. You then allow other proteins to flow through, and this is an excellent purification step that will purify your protein to a high degree. The protein can be eluted by adding an analog of histidine, imidazole, to the solution in increasing concentrations. And this slide shows that imidazole has some structural analogy to histidine, and with increasing concentrations, it will outcompete the histidine of the his tags that are covalently bonded to your protein of interest to allow that protein to be eluted, to allow that protein to be released from its interaction with nickel. So this shows the workflow in this system of DHFR. You receive genetically engineered cells in the mail and grow them up in an overnight culture, and I always have my students do this to save me work and have them get additional experience in the lab. And then you subculture the cells the next day in the laboratory. You grow them without the inducer, monitor their growth with a spectrophotometer, and then at some later time, when the proteins are still in logarithmic growth phase, you add the inducer, and this is a system very similar to the Lactose Operon, and it's induced with the analog of lactose, IPTG. The cells then fill up with protein. You harvest the cells using centrifuge, break them open, and then the proteins are released in order to recover and purify. Once the proteins are purified, because this is an enzyme, they can be analyzed using an enzyme assay specific for this particular enzyme. And also, I always have students run polyacrylamide gels on the proteins, looking at the lysate before the proteins have been purified, and looking at the purified proteins afterwards, so they can get a feel for how well they did purifying the protein. The enzyme assay is an interesting one. Again, it exploits this cofactor that's used along with the enzyme, NADPH, and NADPH can be detected in a spectrophotometer. NADPH has to be detected in a spectrophotometer that has the capability of reading ultraviolet light. Here's the reaction that the enzyme catalyzes, and you detect NADPH, and therefore when the enzyme is in the presence of NADPH, if you monitor the reaction over time, the amount of NADPH diminishes, and that gives you a measurement of the enzymatic activity in your solution. So it's always good to have students work with enzymes because the enzymes can be detected through very specific assays afterwards. I always have the students start with centrifugation protocols, start on the small scale with hand protocols, as I said, but then I always like to have my students use an automated system, use a system to isolate the proteins with professionally packed columns, with pumps to push the solutions through and to change the ionic strength of the solutions. And so in my lab, I have a Biologic LP. The Biologic dual flow is the next step up. What I especially like about the dual flow is that it has an interface with the columns that's very similar to the interface that students will see on the large scale if they get a job in biomanufacturing at a large-scale biotechnology company. And of course, these instruments are expensive, and therefore sometimes schools will team up and buy one for several schools and then trade them around. But it is well worthwhile to have students work with an automated system. Once I've had the students work on this on the small scale, and then I always like to try to generate interest by relating how small laboratories will produce proteins to how a biotech company will produce proteins at the kilogram level by growing cells in 10,000-liter tanks and then isolating 10,000 liters of that harvested cell culture solution, passing it through chromatography columns that are 9 feet in diameter, and then isolating it for purity to generate protein pharmaceuticals. And I always try to remind the students that these are drugs that are saving someone's mother. These are drugs that are addressing diseases. Many of those diseases, before these drugs were developed, there was no treatment. Not no cure, no treatment. That is, if a patient developed one of these diseases, there was nothing that a physician could do for them. After the genetic engineering of the cells, the laboratory research and development, and then the scale up to biomanufacturing scales, these drugs now are available to treat patients and to really change their quality of life. And so I always like to relate to students how this is done in biotech companies. And by the way, this is an excellent career for them. Biotech companies often will pay for their additional schooling, and this is a career that provides a real career ladder where someone who is hired at technician level 1 near the end of their career might be in charge of the company. And that's a real story. There are many examples of people that have done this. The future belongs to biotechnology. Biomanufacturing will be used. Right now it's used for protein pharmaceuticals. Later on it's going to be used for biofuels. It'll be used for producing raw materials for textiles. It will probably touch every component of our lives. And so I try to have the students buy into that excitement over biotechnology. It really is the technology of the next century. Thank you very much for listening, and thank you, Bio-Rad, for inviting me here.
Great. Thank you, Jim. We appreciate your talk. I wanted to share a few resources and references with you to let you know that Bio-Rad has several curriculum training specialists that are available to answer any questions that you might have about any of our products or your curriculum and training needs. The email and our website are listed on the slide in front of you. We also have an excellent technical support group, which is available. They're a well-trained group of scientists ready and able to answer all technical questions you have about biotechnology and products. Also, there's the Northeast Biomanufacturing Center and Collaborative, also known as the NBC2. They also are a great resource. Their website is listed there as well. And they also have special workshops that you can look into called Protein is Cash, which gives you great training on protein techniques. Also, we have BioLink, which we mentioned previously that Jim is a member of. Elaine Johnson is the director of that, and they're a great consortium promoting biomanufacturing and biotechnology throughout the United States. And we've also provided Jim's email as well. He's happy to entertain any questions and comments that you might have for him.
Send me an email, just for fun.
Exactly. Exactly. Thank you. So we also wanted to let you know that the protein expression and purification series will be available this summer. And we have the catalog ordering information number presented there on the left. To give you an overview of this series, it's a modular series. It has 4 different modules, the first being the growth and expression module, the second being SDS page and electrophoresis, the third being the purification module, and the last, the DHFR enzymatic assay module. So the purification module has 3 separate options, the first option being a centrifugation purification method. So for those people who don't have the specialized chromatography instrumentation, all that's required for that would be a good 16K microcentrifuge, and then you could do the centrifugation method. Option number 2 is for if you have specific purification equipment such as the Biologic LP or DuoFlow systems. This allows you to hand pack your own columns and do the purification that way. The third module is using the pre-packed cartridges and using the specialized purification instrumentation. And now we'll take some time to answer any of the questions that you've sent in. Please continue to send them in during this time. And we'll go to the first question that we have. So, Jim, if you can answer this, we have a participant saying, what is the process or method used in folding? I'm assuming for protein folding.
Yes, so that's a really complicated question. And so protein folding isn't well understood, and what biotech companies mainly do is empirical, that they will add their protein of interest gene into a bacterial cell and then find out whether or not it folds. If the protein doesn't fold in E. coli, it often will form this large aggregate called an inclusion body. The inclusion body is good and bad. It's bad because the proteins aren't folded properly. It's good because the inclusion body can be used as an isolation method. Once the cell is broken open, you can spin down the inclusion bodies, which gives you an extra purification step, but then you have to unfold the protein, usually with urea, and then dialyze out the urea and have the protein fold back up. So the protein folding is dictated by its amino acid sequence, but the specifics of how that amino acid sequence ends up with a particular three-dimensional structure with a particular folding of a protein still are being worked out. It's really difficult to predict. There's an interesting website on Stanford that you can run as a screensaver where they're examining protein folding, so that might be of interest.
Thank you, Jim. The next question is, what is the most common first column for purifying proteins in biomanufacturing processes?
So the most common first column will be protein A for antibodies. And antibodies are a huge percentage of the proteins that are being produced at the large scale in biomanufacturing for a biotech company. So it'll almost always be protein A. And that's interesting because there's a rule where you want to use your least expensive resin first so that the protein preparation that your first resin is seeing, that your first chromatography column is seeing, will be least expensive because there might be some gunk in there. And in this case, though, you're using your most expensive affinity chromatography system first. And you can get away with that because antibodies are secreted, and the secretion from the mammalian cell into the harvested cell culture fluid surrounding the cell is essentially another purification. So the proteins come out relatively pure, so it doesn't gunk up your first column. If your columns aren't antibodies, the first column will usually be an ion exchange column. It'll be a positive ion exchange column if your protein is negative, a negative ion exchange column, negatively charged ion exchange column if your proteins are positively charged.
Great, and I think we have just one more question that we have time for, and that is, what is the average number of columns needed to get the protein of interest to 99.99% purity?
For biotech companies, the smallest number of columns that I've seen has been 3, but pretty typical is 5 chromatography columns. So one after another, after another, after another, after another, and the proteins will be purer and purer and purer after each step. And then in the end, you have to prove to the FDA that you have proteins that are more pure than ivory soap, which was 99 and 44/100% pure. And again, quality control will perform several hundred tests maybe to prove along the way that your protein is properly purified. And this is important because you have to have confidence enough that your protein is pure to be injected into children. Remember that proteins, because they're three-dimensional, cannot be taken like a pill. If you took a protein pill, the minute that it met stomach acid, it would unfold and therefore be non-functional. So all proteins are injected directly into the bloodstream, and therefore they had better have the appropriate purity, they had better be absolutely sterile, they had better have the right salt concentration surrounding them in order to function properly and not cause problems in the patient.
Perfect. Thank you so much, Jim, for being here for this. And on behalf of the Biotechnology Explorer program, we want to thank you all for your attendance.
Well, thank you for inviting me.
Thank you once again for joining us. This concludes today's web conference. You may now disconnect. When we close, a survey will appear on your screen. We appreciate any feedback you provide.